Hello, welcome to Biology, the scientific study of life. Again, more about enzymes. How enzymes allow the EA barrier. Proteins, DNA, and other complex molecules of the cell are rich in free energy and have the potential to decompose spontaneously. That is, the laws of thermodynamics favor their breakdown. These molecules persist only because at temperatures typically for cells, few molecules can make it over the hump of activation energy. However, the barrier of selected reactions must be occasionally be surmounted for cells to carry out the process necessary of life. Heat speeds a reaction by allowing reactants to attain the transition state more often, but this solution would be inappropriate for biological systems. First, high temperature denature proteins and kill cells. Second, heat would speed up all reactions, not just those that are necessary. Organisms therefore use an alternative catalysis. An enzyme catalyzes a reaction by lowering the EA barrier, enabling the reactant molecules to absorb enough energy to reach the transition state even at moderate temperatures. An enzyme cannot change the delta G for a reaction. It cannot make an endogenic reaction exogenic. Enzymes can only hasten reactions that would occur eventually anyway, but this function makes it possible for the cell to have a dynamic metabolism, routing chemical traffic smoothly through the cell. And because enzymes are very selective in the reactions they catalyze, they determine which chemical process will be going on the cell at any particular time. Substrate specificity of enzymes. The reactant on an enzyme acts on is referred to as the enzyme substrate. The enzyme bind to its substrate of substrates when there are two or more reactants forming an enzyme substrate complex. While enzyme and substrate are joined, the catalytic action of the enzyme convert the substrate to the product or products of the reaction. The overall process can be summarized as follows: enzyme substrate plus notice that enzyme substrate complex and enzyme products. Notice that, relatively notice. For example, the enzyme sucrose must enzyme names end in AS catalyzes the hydrolysis of the disaccharide sucrose into its two monosaccharides, glucose and fructose. Notice that sucrose, fructose plus H2O, water. The result or chemical reaction sucrose minus H2O complex. Also sucrose plus glucose plus fructose. The reaction catalyzed by each enzyme is very specific, and an enzyme can recognize its specific substrate even among close related compounds, such as isomers. For instance, sucrose will act only on sucrose and will not bind to other disaccharides, such as maltose. What accounts for this molecular recognition? Recall that enzymes are proteins, and proteins are macromolecules with unique three-dimensional conformations. The specificity of an enzyme results from its shape, which is consequences of its amino acid sequences. Only a restricted region of the enzyme molecule actually binds to the substrate. The region called the active site is typically a pocket or groove on the surface of the protein. Notice that. It is usually the active site is formed by only a few of the enzymes amino acids with the rest of the protein molecule, providing amino acids with the rest of the protein molecule, providing a framework that determines the configuration of the active site. This specificity of the specificity of an enzyme is attributed to a compatible fit between the shape of its active site and the shape of the substrate. The active site, however, is not a rigid, respectable, or receptacle for the substrate. As the substrate enters the active site, interactions between its chemical groups and those on the amino acids of the proteins cause the enzymes to change its shape slightly so that the active site fits even more snugly around the substrate. This induced fit is like a clasping. Notice that. A clasping handshake induced fit brings chemical groups of the active site into position that enhance their ability to catalyze the chemical reaction. In an enzyme reaction, the substrate binds to the active site. In most cases, the substrate is held to the active site by weak interactions such as hydrogen bonds and ionic bonds. Side chains are group. Notice that. Um, of a few of amino acids that make up the active site catalyze the conversion of substrate to protect the productive parts from the active site. The enzyme is then free to take another substrate molecule into its active site. The entire cycle happens so fast that a single enzyme molecule typically acts on about 1,000 substrate molecules per second. Some enzymes are much faster. Enzymes, like other catalysts, emerge from the reaction in their origin from therefore, very small amounts of enzyme can have a huge metabolic impact by functioning over and over again in catalytic cycles. Most metabolic reactions are reversible, and as this case, notice that these are reversible metabolic bond. An enzyme can catalyze both the forward and the reverse reaction, which re reaction provides depends mainly on the relative concentrations of reactants and products. The enzyme always catalyzes the reaction in the direction of equilibrium. Enzymes use a variety of mechanisms that lower activation energy and speed up a reaction. Notice that also steps. Also, first, in reactions involving two or more reactants, the active site provides a template for the substrates to come together in the proper orientation for a reaction to occur between them. Second, as the active site of an enzyme clutches the bound substrate, the enzyme may starch the substrate molecules toward their transition state, conformation, stressing, and bending critical chemical bonds that must be broken during the reaction, because EA is proportional to the difficulty of breaking the bonds. Distorting the substrate makes it approach the transition state, and thus reduces the amount of free energy that must be absorbed to achieve a transition state. Third, the active site may also provide a microenvironment that is more conducive to a particular type of reaction than the solution itself would be without the enzyme. For example, if the active site has amino acids with acidic side chains R groups, the active site may be a pocket of low pH in an other ways otherwise neutral cell. In such cases, an acidic amino acid may facilitate H positive transfer to the substrate as a key step in catalyzing the reaction. A fourth mechanism of catalysis is the direct participation of the active site in the chemical reaction. Sometimes this process even involves brief covalent bonding between the substrate and a side chain of an amino acid of the enzyme. Subsequent steps of the reaction restore the side chains to their original state, so the active site is the same after the reaction as it was before. The rate at which a particular amount of enzyme converts substrate to product is partly a function of the initial concentration of the substrate. The more substrate molecules are available, the more frequently they access to the active sites of the enzyme molecules. However, there is a limit to how fast the reaction can be pushed by adding more substrate to a fixed concentration of enzyme. At most point, the concentration of substrate will be high enough that all enzyme molecules have their active sites engaged. As soon as the products exist in an active site, another substrate molecule enters. At this substrate concentration, the enzyme is said to be saturated, and the rate of the reaction is determined by the speed at which the active site can convert substrate to product. When an enzyme population is saturated, the only way to increase the rate of product formation is to add more enzyme cells. Sometimes do this by making more enzyme molecules. Okay, notice the induced fit between an enzyme and its substrate. This notice that model, graphic model, substrate, active site, notice enzyme substrate complex here. Notice also the active site and catalytic cycle of an enzyme. Notice substrate. This type substrate interactive site enzyme changes shape, so its active site embraces the substrate induced fit. Notice that substrate. Two substrates held in active site by weak reactions such as hydrogen bond and ionic bond. Notice 3 also active site and R group of its amino acid. Acids can lower EA and speed up a reaction by acting as a template for substrate orientation, stressing the substrates and stabilizing the transition state, providing a favorable microenvironment, participating directly in the catalytic reaction. Substrates, for notice that substrates are converted into products. Notice these are products are released here, and the enzyme active site is available for 2 new substrate molecules. Okay, thanks for listening.